part two of our discussion of the practice questions for this week. You see a problem that's a little familiar. Actually, it's a little simpler than the problem that you had for the performance exam, but it works on the same lines. And I did have a couple of people who asked for another problem that was like this, and so I specifically went out and got one, just because of the fact that this particular lab deals with a lot of different concepts, and so I think that it stands a pretty good reason that the lab question that you do could very well end up being a lab dealing with Drosophila breeding. So you need to make sure that you're understanding how to go about looking at this type of problem. But I wouldn't expect it to be a problem where you could look at it and see straight away, okay, you see white eyes, and the first thing that most of you should at this point think is that's a sex-linked trait. And so it's not going to follow the simple rules of Mendelian genetics, but it actually does follow Mendelian ratios once you place it in the right spot. All right, so let's take a look at what we have in this particular problem. In fruit flies, the phenotype for eye color is determined by a certain locus. Capital E indicates the dominant allele, and lowercase e is going to be the recessive allele. We have the cross between a wild-type fruit fly and a female white-eyed fruit fly produced the following offspring. All right. Once you go through both of these, the ultimate goal here is to determine the genotype of the original parent. So let's see what we can write down. Looking at E and lowercase e, one of the things that we could possibly do, that wild type would be our dominant, and then the white eye that they're talking about is going to be the recessive. So we could set this up as in two different ways possibly. Let's see what happens if we set it up specifically as a straight cross. We're only dealing with one characteristic here, so this is going to be pretty simple. We have the wild-eyed male. Let's see, what do we? Yes, the indicator, the cross between a male wild-type fruit fly. So if we said that he had two of the dominant genes, and we're going to cross it with a female white-eyed, well, if we're going to say that white-eyed here is a recessive characteristic, then we would give it two lowercase e's. And notice that we're just dealing with one characteristic. We only have one set of characteristics here. Well, just looking at this cross, this is going to result in 100% of our offspring being heterozygous. So 100% would be heterozygous in this format. Well, if we did say that wild type is the dominant, then 100% of our offspring would be phenotypically dominant in this case. And that would make this the white-eyed. Well, if we reverse that and made white eyes the dominant characteristic and wild type the recessive, still 100% of our offspring would be heterozygous. Either way you cut this, it would be heterozygous. And what you would end up with is 100% of your offspring showing the dominant, showing whatever the characteristic is that's dominant. Another possibility here is if the male that's wild type were heterozygous. If the male is heterozygous, because it doesn't say that it's true breeding or anything, so but one of them has got to be true breeding if it's showing the recessive characteristic. So if we make one of them heterozygous and the other one homozygous recessive, what will this do? Well, this is going to give us that 50% phenotypic split. So we would say half of these would have wild type, and half would have the white eyes, and that would be true whether we said that white eyes was dominant or recessive. So in these two cases, these two possibilities, either 100% of our offspring are going to be phenotypically dominant, or what we're going to have is a half and half split. Well, let's look up here at the top. Our F1 generation, I have two different types here. Well, I've also got this brown-eyed female down here, and I'm not really sure where this is going to come into play because there are no brown eyes in this next generation down here, so this could have just been, not even sure where that particular piece of data came from. Possibly they were trying to show that it was a mutation. So 45 and 55. Well, wild type, white eyes. So if we were only looking at wild type and white eyes, and we don't pay any attention to the sex of the flies, then this could be a viable option. We could have had one parent that was heterozygous and one parent that was homozygous recessive. That's a possibility. But at this point in time, we know that white eyes are actually a sex-linked characteristic. If we look at this as a sex-linked characteristic, then the way that we would take a look at this is by just remembering, okay, so you have the male and the female, all right? Well, the male is wild type, so he's going to be showing wild type, and the female is white-eyed. Well, let's just say that white eyes is a recessive characteristic, so I'm going to put the lowercase e next to each one of these X's that came from the mother, and since the father, or the male fly, is showing the dominant characteristic, or is he's showing the characteristic of being wild type, I'm going to put a capital E next to his X chromosome, because we know that in a sex-linked trait, you're going to have the it's going to be riding around on the X chromosome. So if we cross these, here's the male and the female. Well, what's going to take place? Here are your females. They both have one X chromosome with the dominant gene and one X chromosome with the recessive gene. Now what about the males? Well, look at this. All of the males are going to have the recessive gene, because if the mother is showing that recessive characteristic, that means that both of her X chromosomes have that recessive gene, which means that she will only be able to give that recessive gene having on the X chromosome to all of her offspring in the gametes. So that means that every son that she has is going to have an X chromosome with that recessive gene on it. So this would mean that 100% of the females would be, well, we've got the dominant characteristic and the recessive characteristic here. So whatever, they would be phenotypically dominant, whatever that is. And 100% of the males would show the recessive characteristic. Well, let's look up here. We have 0 wild-type males. We have 0 white-eyed females. We have 55 white-eyed males and 45 wild-type females. Well, we said that this could be a possibility here. Half of them would be showing the dominant characteristic and half would be showing the recessive characteristic if one were heterozygous and the other is homozygous recessive. But this doesn't have anything to do with the sex of the flies. If this were the case, what should we see up here? I should see as many wide-eyed females, or I'm sorry, whatever this one is. Let's say that the dominant characteristic were wild eyes. I should see as many female with wild eyes as I do males with wild eyes, and I should see as many females with white eyes as I do males with white eyes. So regardless of which of these two, which one you set as your dominant and which one you set as the recessive, you're going to see an equal number between males and females of this characteristic. Well, is that what we see up here? That is not what we see here. Only the males are showing this mutated white-eyed gene. The females are all showing the wild type. Well, look down here. 100% of the males are one phenotype and 100% of the females are another phenotype. Well, I think that that's probably going to be what our answer is then. Take a look. Out of this, we have 45 and 55. We would expect 45, we would expect half of our offspring to be wild type females, if we say that wild type is the dominant, and we would expect for 100%, or in this case there's 100, well 101 if you count this one out here, but 100 of our, out of 100, 50 of them should be white-eyed males. So all of our males should be white-eyed here. Well, look down here. All of our males are showing the recessive characteristic. Therefore, this is going to be my hypothesis, at least, for what our genotype for the parents are going to be. So between these two, if we said this was a possibility and that's a possibility, the data supports this a little bit more than it does this. And it supports this more because this has the phenotypes separated by sex. This does not. This we would expect for our offspring to be equal numbers of male and female between each of these two. Now, half are going to be this phenotype, half will be this phenotype. But we should see that there's no difference between males and females. And here, there is a definite difference between males and females. So, if we wanted to do a quick check of this, just to see whether or not it follows Mendelian genetics the way that we have drawn it out here, obviously we expect 100% of our females to be white-eyed or wild-eyed, and 100% of our males to be white-eyed. And out of 100 offspring, I would expect 50 to be each one. Well, what do I see up here? I see 45 wild-eyed females, and I see 55 white-eyed males. Well, a quick run of chi-square, and what I end up with is a chi-square value that is actually equal to 1, and with 1 degree of freedom at the 5% level, I get 3.84. 1 is below that, so I get pretty much exactly what I expected here. So this follows the model. So it follows what we expect. It follows the model. Okay? So that at least takes care of the first generation. So let's see if the second generation follows along with our hypothesis. Alright, so if we follow along, the wild type and white-eyed individuals from the F1. Well, look at the F1 up here. Our only wild type are female, and our only white eyes are males. So let's see. So if here's our female, and they're all showing wild type, but they're all heterozygous, right? They're all heterozygous because, look from here we go, right here. All of our females are heterozygous, so that means we have this female, and we're going to cross it with a male who's got white eyes, so he's carrying the recessive gene. And that means that he's white-eyed. So here is our female, and here's our male. So if we cross these two, what are we going to get? Well, when we cross these, you're going to get, let's put it into a square and find out. Here's the male, and here's our female. Well, look what we have here. Capital E, lowercase e, that's a female, so that would be phenotypically wild. This, the X chromosome from dad that has the recessive, and the X chromosome from mom that's got the recessive, and so that's a double recessive, which means that this one is going to show the recessive characteristic, so that will be wide-eyed female. Here is the normal gene, or the dominant from mom, and the Y from dad. So this would be a wild type male. And then here is the recessive gene from mom and the Y from dad. So that would be a wide-eyed male. So we have wild-eyed male, wide-eyed male. Well, all four of those are different, which means that we would expect a fourth of our offspring to be wild-eyed female. We would expect 1/4 of our offspring to be white-eyed female. We would expect 1/4 of our offspring to be wild-eyed males. And we would expect for 1/4 of our offspring to be white-eyed males. So we would expect essentially a 25-25-25 split among all of those. Well, if I've got 23 and 31 plus 22 plus 24, so that gives me 100. Notice that I have 0 brown-eyed females. Don't even know why the brown-eyed female was up there. I would expect then to get 25, 25, 25, and 25 of each one of those. Well, if I expect this and I observe that, what I end up with is a chi-square value of 2, exactly 2. Well, at 3 degrees of freedom, at 5%, the magic number for chi-square is 7.81. Well, 2 is less than 7.81, so we fail to reject the null again, and so this also follows the model. Since this one follows the model, and I used the genotypes from my F that I hypothesized from my F1s, together, this is 2 really big pieces of evidence that we have that in all likelihood is the phenotype or the genotype of the initial parent. So let's just following back along. We only had one characteristic. If we had had more than one characteristic, which I would expect for you to have, you're going to have to do your probability when it comes to putting your gametes together. Okay, thinking about each characteristic as its own separate Punnett square, finding out what is the probability that you'll have each one of those for, and then putting that together with the probability from the other characteristic. So if I were coming to a final answer for this particular question, then I would say here is the genotype for the initial parent. That white eyes is a sex-linked characteristic. A sex-linked characteristic that is, of course, course, riding around the X chromosome, and it is recessive. So the male in this case has the wild type gene, the female had the recessive gene. And there we go. Rather than going to a third video to do the bonus questions, we're just going to go ahead and quickly do these two bonus questions. I did these are not even in the format really as the questions that you will see on the AP exam. So I went ahead and just put them on here because I would make sure that I knew what was going on with these, because you might have to put this together conceptually with something else in order to write an answer. Describe 3 differences between mitosis and meiosis, and then down here 2 events that are common in both mitosis and meiosis that ensure the resulting daughter cells have the appropriate number of chromosomes. So what I would do is think about making a chart of all of the things that mitosis and what are the differences and similarities with mitosis and meiosis. So let's deal with the differences first. So I'm just going to come down here and I'm going to make a chart, and I would make sure that I knew that I could quickly and easily recall all of these differences between mitosis and meiosis. Alright, so to start with, let's just go from the very beginning on what our general purpose of mitosis and mitosis are. In mitosis, you know that you start out as a 2N, it goes to 4N, and then it splits, and then when you're finished, you're back at 2N. In meiosis, however, we start at 2N, it goes to 4N, and then to 2N, and then to 1N. In other words, we go from being a diploid cell to being a haploid cell, whereas in mitosis, we go from being diploid to still being diploid. Mitosis represents genetic continuity, because you are making, every time a cell goes through mitosis, the result is genetically identical cells. Theoretically, you might have some mutation, however, they're supposed to end up with genetically identical cells because it's preferable to have the same DNA in all of the cells of a multicellular organism. Whereas in meiosis represents genetic diversity, because you end up with 4 genetically unique gametes that are haploid. In mitosis, you have only one cellular division process that happens. In meiosis, you have 2 cellular division processes. Mitosis is part of the cell cycle. So mitosis is a cycle. It runs essentially continuously. Now, we did learn a little bit of in one of the earlier units that you know that in mitosis you do have several checkpoints, and that for the most part cells, most animal cells particularly, are in the G0 stage, and so they're not actually continuing through the cycle. But each one of those cells can again go through a cycle. So you have a cell, it goes through the cycle, and then you have 2 cells, and then each one of those can go through the cycle and you have 2 cells, and so on and so forth. Whereas meiosis is a process. Once it is ending in our gametes, so it produces gametes and it's done. It does not continue. Once you have gametes that are haploid, they are not genetically identical to the cells that we started with, and they're done. Each one of those does not typically go through mitosis. Now, there are some cases where you can end up with cells going through mitosis, and that's going to depend on the difference in, especially in plants, when do they have the sporophyte and when they have the gametophyte part of their life cycle. But for the most part, once this is done, it's done. In mitosis, think about the chromosomes that come together. The chromosomes that come together in mitosis are only sisters. So sister chromatids come together. Whereas homologs or homologous chromosomes do not come together. In meiosis, the homologs do come together, and it just so happens that in prophase 1, when we have homologs coming together, we actually have the tetrad. Remember, you have the two homologs plus each homolog has its duplicate or its sister. So we have this tetrad. It's the only time you have a tetrad anywhere in this process. So during prophase 1, metaphase 1, and then at the beginning of anaphase 1, you have this tetrad. And then during anaphase 1, what's pulled apart here? Well, homologs are pulled apart. And it's in anaphase 2 where the sisters are pulled apart. Well, what else is going on during that prophase 1? Meiosis has crossover. Remember that wonderful process that mixes up the genetic material. Mitosis has no crossover. Mitosis, what did I just say about being split apart during anaphase? Sisters are split during anaphase. Over here, the tetrad splits during anaphase 1, and what splits in this tetrad, the first one, is the homologs, and that's in anaphase 1. The sisters do separate, but that takes place during anaphase 2. And remember, by that time, they're actually not technically sisters anymore because they're genetically unique thanks to crossover. Finally, a difference that I would want to mention is that mitosis is for growth and repair, to replace old cells, whereas meiosis, the purpose of meiosis is to produce gametes for sexual reproduction. So you have a lot of differences here. You have a lot of differences to choose from. If you had this question right here, write down 3 differences. You could take any three of these things that you see here and give some specific information about that. You could actually give more. All right, so that's our first one. I wrote that way down here just because of the fact that it takes a lot more. Now, describe 2 events that are common to both, and look specifically, what are we talking about, that ensure the resulting of daughter cells so that inherit the appropriate number of chromosomes. Well, think about when things get pulled apart to go to either end. Things line up during prophase and they're pulled apart during anaphase. Well, every time they line up, think about what's lining up. In mitosis, you have sisters line up. And how many is that? It's a pair, right? So pair. Well, during meiosis, what lines up? The homologs, which is a pair, even though, remember, they code for the same things, but they're not genetically the same because one came from each of your parents. And then each of the homologs has its sister or its duplicate. So still, you have a pair. And it just so happens that in meiosis, the pair is the homologs plus their duplicates. So every time we line up in pairs. So when we line up in pairs, half can go in one direction and half can go in the other direction. So whether you've got one pair, as in mitosis, or two pair, as in meiosis, either way, during anaphase, those chromosomes are going to get pulled apart. If it's in mitosis, it's simply anaphase and they're pulled apart from their sister. If it's in meiosis, in anaphase 1, you get 2 going to that side and 2 going to this side. And in anaphase 2, now then we go back and we split the sisters. And remember this thing called non-disjunction, which could happen here, and that could easily be another part of this question. What would happen if you don't have that split? Well, you have something called non-disjunction, and so you'll end up with some gametes with too many chromosomes and some with too few, and that's obviously going to be a genetic mutation, a chromosomal genetic mutation. So there you go. 2 events that will assure that chromosomes, the right number of chromosomes go to each cell after division, and then a list of things that are different about mitosis and meiosis. Given all of these things, think through, what are the units, what units would you like to see a complete review for, or a little bit more of a review that you would like for me to do. Send me a text on Remind, send me an email, or put it in the discussion block. Be prepared to do your mock exam next week, and I'm going to give you also, before the end of this week, a set of some review materials for the final, the multiple choice final. Although you already have the outline and whatnot, so that's about it. Let me know if you have any questions.